These PowerPoint slides are a lot. There's a lot of information on here. So I recommend that you have these with you in some fashion when you come here. You have like your computer out. I've done this to you because it's the first day and we're starting already. But I won't be doing it moving forward. This is not required at all, but you do this and not an excuse for you to buy a printer. This will certainly kill your printer. If you do end up doing this, that won't show it.
Yeah.
Yeah, can I use your tablet?
Yeah.
Great. You want a copy? Good. If you're not accustomed to printing PowerPoint slides, I want to show you this little thing here. Be sure you don't just go print and get it going. You can't do that. That'll blow up your printer. So you go to handouts. This is how you print it, right? Usually you print 6 slides per page because these are all the same size regardless. And, sometimes it's impossible to see what's on here because of the type font size, but it just gives you a good way to kind of, take notes and pay attention. And but it's up to you. However you guys study is good with me. Okay, so let's get into, if there's no more questions. I think I covered everything. Real quick, so Monday, you guys have lab, right? So what do you have to bring to lab? Notebook, right? And then do you have to bring printouts or anything? No. So I'm going to give you guys printouts for the first day. It's pretty straightforward lab. We're just doing pipetting and I think we're analyzing agarose gel, stuff like that. If you get a chance, you want to read through the packet, it's not a bad idea. It'll kind of make things work more quickly, but it's not something that I'm requiring or going to keep up on. All right. Okay, chapter 1, called The Origin and Evolution of Cells. If you have an opportunity to take evolutionary biology in your study as a bio major, it's an awesome course. Definitely one of the more interesting courses that I took when I was getting my degree. This book does not get into it too bad. This is very introductory in terms of this text. But it does touch on a little bit, where could have these cells come from that we're going to talk about, right? So, any A&P people in here? Anybody taking A&P? So what is the most fundamental way to break down anatomy and physiology? It's 2 words. I think you said it. Structure and function, right? So how something is built starts to give way into understanding how something works. Cell is, in a lot of respects, the same way. How a molecule is built is going to kind of indicate how it works. And that's why cell biology and molecular biology are so tied together. Right? So it's really understanding the molecules, how they're built. Now you can start to begin to understand how they function. So where did these molecules come from, right? Where did these organic molecules come from that use carbon as the basic building block within them? So a long time ago, life started to emerge, but obviously the building blocks for that life needed to emerge earlier on, right? So when we talk about prebiotic conditions, right, we talk about conditions before life was around. So the environment of the earth prebiotically was very different. You had high levels of carbon dioxide, nitrogen, hydrogen sulfide, and carbon monoxide. So a pretty toxic environment, something we wouldn't be able to survive in that environment today. I mean, it's like raining acid rain. You got volcanic activity all over the place. It's a highly reducing environment, right? So reduction is a type of chemical modification that we're going to talk about. The opposite would be oxidizing. But from this like primordial soup, you started to generate more life amenable compounds, right? And the Earth actually formed into an oxygen-rich environment. A study that was done early on by Stanley Miller, and it's kind of a sloppy experiment in my opinion, but kind of worked, is he set up this glass apparatus where he kind of mimicked the conditions of the early planet. So he had the chemicals, the inorganic chemicals that were in there that were present on Earth at that time. He added heat, right, to indicate, just either warmth from the sun or volcanic warmth. He had electricity in there to indicate, simulate lightning, a cooling effect from the upper atmosphere. And he refluxed this thing for weeks, right? He just let it cycle around and around and around. And after a few weeks, he got this black goop that built up in the bottom of that system right there. And he started to analyze it. And what did he find? Well, most importantly, he found the presence of amino acids. So amino acids were able to almost spontaneously form from just these basic inorganic chemicals. Now that's not enough, right? Just having amino acids isn't going to do it. In order to have a living system, you have to have more complex macromolecules, which are large molecules. So the assembly of all these amino acids together, now you can start to get a protein, you can get a functional molecule. And how that process, evolved is up to a lot of debate. But at least this explains early on where these organic compounds could have come from. As cells started to develop, you need to have some sort of way of information passing down from parent cell to daughter cell. Just water my gut. So, what's the information molecule that we know of today that provides all the genes for us? DNA, right? Well, DNA is more stable than its sister molecule, RNA. So it makes a really good molecule to store the blueprint of information. But it can't do anything on its own. It's not enzymatically active. You need other molecules, enzymes, to manipulate the DNA if you either want to copy it or if you want to read it. So its sister nucleic acid, though, ribonucleic acid, or RNA, is a little different. RNA can contain information in it because it's made up of nitrogenous base pairs, which make up the code of the RNA molecule. But RNA can also form spontaneously into different shapes and conformations. And these conformations can actually make it intrinsically catalytic. So RNA molecules, some of them, can act as enzymes and also contain information within the molecule itself. And that's why we term them ribozymes, because they're information molecules that can fold up into these different forms, and they are intrinsically, which means like just by themselves, catalytic. So it's pretty obvious that the first information molecule was in fact RNA. And then over millions of years, that started to get replaced with the more stable DNA molecule. But RNA is still used in our cells. And a lot of the, cell biology is not a new field, right? But a lot of the molecular characteristics of all the different components of the cell have only been discovered within the last 40 years or so. And RNA is one of them. So the ribosome, for example, ribosome is 50% RNA and 50% protein, and a lot of that RNA within the ribosome is catalytic. It's actually what produces the peptide bond when it's building proteins. So it transferred over to DNA, and we term these genes, these are segments of DNA that can code for either proteins or RNA molecules. The definition of a gene used to be a segment of DNA that coded for a protein, but that's not always true. Some codes of DNA code for RNAs and then stop there. right? There's a whole bunch of different RNAs that function in the cell and have important functions. DNA is able to exhibit, both molecules, sorry, are able to exhibit what's called complementary base pairing, right? So A's bind to T's, C's bind to G's, right? And they bind through a special type of bonding called hydrogen bonding. It's like a weak bonding, which means that it's enough to keep them together. But if the molecule needs to unzip or come apart, it can do that. So this is an important property for the molecule to be able to replicate itself. In other words, copy itself. Because now if you have a double-stranded molecule, you could separate it. And if you read one side of the molecule, then you know the other side of the molecule because of complementary base pairing, A's to T's, C's to G's. Additionally, because you have 4 different nucleotides that make up DNA, you can access information from that, right? So different sequences can ultimately lead to different shapes and designs of proteins. And that architecture is ultimately what's really important in providing function within the cell. All right, having that complementary base pairing is what enables us to read the DNA and then produce a molecule of RNA. That process is called transcription. And it occurs where the DNA is. So in eukaryotic cells, it occurs in the nucleus, and prokaryotes it just occurs in the nucleoid, or where the DNA is. And then that RNA has to travel somewhere else in the cell, specifically to a ribosome, to be translated into protein. So this is called the central dogma of information because it's the flow of how information moves in the cell. It goes from DNA to RNA to protein. You have to have this message system enclosed or encased in something, or you just have a chemical floating around in a pond or something like that. So you need to have something to segregate the cell from its environment. These are membranes, and membranes are formed by phospholipids. All right, so now we're talking about a lipid molecule, and if you know anything about lipids, like the fat from your steak, it's very difficult to clean out of the pan, right? Because it doesn't dissolve in water. It's hydrophobic. Now, those fat molecules are completely hydrophobic. Phospholipids are like a modified version of a triglyceride, which is your fat molecule. And it actually, part of the molecule is hydrophobic and part of the molecule is hydrophilic. So if this is a triglycerides with your 3 fatty acid chains and your glycerol moiety up there, a phospholipid looks more like this, contains phosphate here. And the addition of this group, which is polar, and we're going to get into that in the next chapter, changes the physical properties of the molecule. So that one end of it is attracted to water, or hydrophilic, right? Philic in Latin means love. And the other side of the molecule repels water, is hydrophobic. This is hydrophobic here. This is philic. So if I had a handful of these molecules and I threw them into a glass of water, there's only one way that they can orient themselves. They'll start to scrunch up tails to tail like this. And they'll do that continually until they form a sphere, because we have an aqueous environment here, an aqueous environment here, which makes the heads of this molecule happy. But again, these things are scrunching together in this area because they're repelling the water. So here you have a molecule that has 2 different physical properties on it, and now it's orienting itself into a shape and a structure that can segregate, the environment itself. And they can do this spontaneously. So as long as you had some RNA floating in the pond and you had a bunch of phospholipids floating in the pond as well, this thing by chance could have formed a living cell. Okay. So these membranes end up being 2 layers, right? It's a bilayer. Call it a lipid bilayer. It's got to take 2 of these amphipathic molecules to scrunch up next to each other. And we call the hydrophilic area the head and the hydrophobic area the tail, right? They always line up tail to tail, hydrophobe to hydrophobe. There's no water in here. There's water on the outside and inside. That would have been the first primordial cell, right? You had your catalytic RNA inside, which could do some stuff, maybe direct the cell to at least divide and do some simple metabolism. And you had a segregated, something that segregated it from the environment. All right, talking about metabolism. The cell has to have a way of providing energy needed to that RNA, right, to carry out chemical reactions. You can't just have a static bunch of molecules sitting in there. There's a dynamic interface that's occurring where these molecules are interacting with each other and changing. You know, one requirement for life is diversity and complexity, right? Without those two things, a living system will fail. That's typically why the more complex, like, or at least oftentimes the more successful an organism can be, and the more diverse the molecules you have within the cell, the more successful. In order to process energy, you have to have a lot of different molecules doing it. And the way that energy, evolved, or the process by which energy is metabolized, evolved in basically three different events over a period of time. So, early on, the Earth was deficient in oxygen. So glycolysis is a process by which you can produce a little bit of energy without the presence of oxygen, right? Basically, you take sugar, glucose, and through a series of reactions, you break it down into pyruvic acid. And that process actually requires a little of this molecule called ATP, but it makes a lot of ATP as well. In fact, it makes more ATP than it uses. So as long as you have like a constant supply of glucose, right, and a way to process some other molecules that are involved in it, which I'm not going to talk about now, you can constantly create ATP. So this molecule ATP, adenosine triphosphate, or adenosine 5' triphosphate, most people just call it ATP, is an electron carrier, right? That'll make more sense in the next chapter, but it's a manner of transferring energy from one reaction to another. Presents a way to couple reactions as well and make them energetically favorable. It's also called energy currency, right? So you think about how fast you blow through the cash in your wallet throughout a day. The cell blows through ATP very quickly as well, right? It will use it like it's burning money, but it has to make enough money, right, to sustain its bank account. So it's a very fleeting molecule. It's made up quickly, dies off quickly. If, an organism is suffocating, right, oxygen gets depleted, and that quick depletion of oxygen, the cell starves of ATP very quickly, and that's why the cell will die starved of oxygen. But you don't need oxygen to do glycolysis because there's none around. The next type of metabolism that evolved would have been photosynthesis. With photosynthesis, you just need a carbon source and light energy, and you can make oxygen as a waste product. So photosynthesis must have evolved before the last one, which is oxidative phosphorylation. And in oxidative phosphorylation, you have a process that uses oxygen, it uses metabolites of glucose, and it can produce a lot of energy. All right, so what do we mean by a lot? Well, glycolysis, when it metabolizes one molecule of glucose, makes a net of 2 ATP molecules. With oxidative phosphorylation, depending on the system, it makes anywhere between 36 and 38 ATP molecules. So it's a lot more efficient of a system, right? These are very elaborate chemical pathways. I'm sure you studied them in Bio 1, and we're going to study them again in here. So now you have a more complex cell. You have cells that have information. They have some enzymatic activity. They have a membrane, and they also have a metabolism. They have a way to process energy. And at this point, cells started to diverge into different cell types. So the major cell type division in cell biology are the prokaryotes and the eukaryotes. What are prokaryotes? Anyone take it micro? Yep, you're studying prokaryotes mostly in that class, right? The bacteria. Eukaryotes are the cells that make up animals, plants, and things like that. Okay, so more complex. All right, so here's these three pathways right here. You can see glycolysis starts with sugar and it makes pyruvate, just a little bit of energy. Photosynthesis, you got CO2, light energy, and you can make sugar now. You can make a food source. And oxygen as a waste product. So now the Earth started to build up oxygen in its atmosphere, and cells could evolve this other method of creating ATP called oxidative metabolism or oxidative phosphorylation. And this generates a lot of ATP just per one glucose molecule. So this is something that could sustain life of a eukaryotic cell, a complex eukaryotic cell that has a lot of enzymes that need a lot of ATP to do the work. So how do you explain the differences between these cells? Well, prokaryotes, typically when you hear prokaryote, you think bacteria, and that's true. They're smaller, all right? They lack a true nucleus. They lack membrane-bound organelles inside of the prokaryote. So they don't have a nucleus. That means the DNA is not segregated in a compartment, and we call the area that it's kind of located and held together the nucleoid. These prokaryotes do have a plasma membrane. All cells have to have a plasma membrane, and they have to have ribosomes as well. Ribosomes are the protein factories of cells. All right, all cells, what do they have? They have the macromolecules, and they have these three things. Whether you're prokaryote, they have plasma membrane and ribosomes, whether you're prokaryote or eukaryote. All right, prokaryotes are divided into two domains. We've got domain bacteria, which is like all the bacteria that we're familiar with today. They include pathogenic bacteria. They're all over the place. And they're just common day present organisms, Staph aureus, things like that. However, obviously these prokaryotes started existing very early on in terms of the age of the Earth, right? When the environment was very different. So the archaic bacteria are typically the cells that today live in really extreme environments, like in a mammal's gut, away from the sun and in another organism, or like in the boiling springs of Colorado. You've got springs that come out of the ground that are boiling temperature. How could anything alive exist in there? Well, that's not true. There's abundant amounts of bacteria that survive in those areas. And it's actually the archaic bacteria which diverged into eukarya. Okay, so of the two, archaea, the more kind of complex and weird bacteria, is the one that diverged into the eukaryotic cells. Cyanobacteria is an important group of prokaryotes because it has the ability to do photosynthesis. Cyanobacteria are one of the most abundant, not the most abundant organism on Earth. And it's found in the oceans, and it provides a majority of the oxygen for the planet, which is very concerning about, global warming. As the temperature of the oceans rise, what's going to happen with this cyanobacterium? And so here's typical, probably E. coli cell, I think. And this is prokaryotic, right? So no membrane-bound nucleus, but it does have a plasma membrane. Instead, that DNA is just like in a central region of the cell that we call a nucleoid, held together by proteins. And this cell is obviously starting to divide, right? So the ability to divide is an important aspect of life, to grow and to spread. And, eukaryotes have a very organized system of cell division, but prokaryotes much simpler. It's just called binary fission. And basically, the DNA needs to copy, and then the cell just kind of splits apart. Prokaryotes also have the presence of these protective cell walls, right? So prokaryotes need to have a protective cell wall. They wouldn't survive with just a membrane. because typically they're in weird environments, like, they're sitting on the handle of a gas pump, right? And rain is pouring on that, right? That's a sort of solution that would cause the cell to pop, right? These cells need to be in isotonic environments. If they're in hypotonic environments, like very aqueous environments lacking salt, osmosis will cause water to go into the cell and eventually it will lyse or blow up. So you need to have that protective cell wall to protect them from osmolysis. So eukarya diverged as a result of archaea, and we'll see how that happened a little bit. And the eukaryotes are more complex. They contain membrane-bound organelles. And as we go through this course, one big focus in cell biology is on the organelles and how they function. And we're going to talk in depth about all of these. The only one that I'm going to skip over, and it's just personal preference, I'm not a plant guy, so we're not going to talk about chloroplast much. We're not going to do any photosynthesis. If you're a plant guy, I completely respect that. And I do like photosynthesis. Gen Bio, I covered a lot, but I just don't feel like covering it this semester. So, honestly, though, it's really the same thing as mitochondrial metabolism. So you're going to be fine for your standardized exams. But we're going to go through these, all of them. Lysosomes, they provide digestive function. They break down molecules in the cell. We'll talk about the ER, its role in synthesis, whether it's the smooth ER synthesizing lipids or the rough ER synthesizing proteins, because the rough ER has those ribosomes embedded in it. We'll talk about that weird word, the Golgi apparatus, and we'll talk a lot about cytoskeleton as well. So it's important to understand the difference between these prokaryotes and eukaryotes, right? The eukaryotes are larger, upwards of about 100 micrometers. 100 micrometers, if you have really, really good eyes, you can actually see that. It's about the size of a period on a page. They have the membrane-bound nucleus, membrane-bound organelles, right? They have a lot more DNA. So prokaryotes, when you see times 10 to the 6, what is that? Millions, right? So millions of base pairs in a bacteria, but almost billions, right, in a eukaryote. So a lot more information to provide all that complexity. The way the chromosomes are organized and how they appear as well is different. Prokaryotes have just one chromosome, and it's circular. Eukaryotes have multiple chromosomes, and they're linear. Right? So these are important differences between prokaryotes and eukaryotes. All right, here's like your generic cartoon of a eukaryotic cell, both a plant and animal cell. I'm sure most of you can probably recognize all these structures by now. When was the last time you took Bio 1? Both you guys, I don't have any ancient people in here, so how long? 8 years. Okay, that's a little long. So, how, I want to be honest with you, man, how well you do in this class can be very associated with how long ago you took Bio 1 and how did the first part of Bio 1, not the second part, the cellular processes. All of this should be a review for you, but I understand if it's not. So if it's been a while since you've had Bio 1, you may need to work a little harder at the beginning of the course, but after middle of next lecture, we're going to start pretty much at that point getting into all new material for most everyone in here, unless you've taken my genetics or taken a genetics course. Most everything we're going to do is relatively new. So, spend the next couple of weeks brushing up on your Bio 1 material if you need to do that. Okay? There's all the structures. Pretty picture. So how did these eukaryotes come into being? Well, it was basically the interaction between two cells, right? You had a archaic cell, which is from that, which is its own domain, right? Engulf a bacterial cell. Remember, these are both bacteria, but bacteria is a separate domain, right? It's the bacteria we're familiar with today. So that bacteria probably had the ability to do oxidative phosphorylation. The archaic bacteria could only do glycolysis. So when it brought that other cell in, that now became the mitochondria. And we know that today because that single chromosome, that circular chromosome that's present in the bacteria cell, is still in our mitochondria today. Right? We have DNA in our mitochondria. It's called mitochondrial DNA. It's very useful in paternity studies and evolutionary studies. And likewise with the chloroplast, an archaic cell ended up engulfing like a cyanobacteria, and that bacteria ultimately became the chloroplast. When you're looking at cell size of eukaryotes, the mitochondria are almost the size of bacteria, whereas this eukaryotic cell is a lot larger. So this process by which, a non-oxidative cell engulfed an oxidative cell, we call this the endosymbiotic hypothesis or endosymbiosis, gave rise to eukaryotes. All right, so you can see eukarya ended up diverging into the most diverse group, and really, the most important group, the group that we're in today, animals. Right? But here's your bacteria over here, like Staph aureus. Right? Here's your archaic bacteria, like your E. coli in our gut. And here's everything that eukarya diverged into: plants, animals, fungus, protists. And protists are kind of a weird group that almost don't fit into anything else. They're single cellular. It's like your paramecium and your amoeba that live in pond waters. Okay, so again, the key thing to note here is that eukarya came from archaea, but there was an assimilation from the bacterial through endosymbiosis that enabled it to diverge and become more complex. All right, everything we've talked about so far is single cell organisms, but now multicellular organisms evolved from associations between unicellular ones. Right. So what organisms are we talking about? Well, the yeast, still single cellular, interactions between that and maybe a protist can start to form multicellular organisms. Right. So here's yeast up there budding. Your paramecium would be a protist. And this Cladomonas represents green algae, which is a separate kind of domain. So when you look at like the numbers of genes in an organism, that can only be so informative. Usually, one thing it will tell you early on is if it's an organism you want to study or not, because the more genes you have, the more work you have ahead of you, and determining like what numbers of non-coding genes you have is going to make an enormous amount of work. But the number of genes you have does not indicate like complexity or anything like that, or the number of chromosomes as well. A potato, for example, has, I think, 127 chromosomes, something like that. How many chromosomes do we have? 46, right? So, chromosome number and gene number are not indicative of complexity all the time. Okay. All right. So why don't we take 5 and then we'll get back into, and when I say 5, I mean really 5, because I don't want to be here all day, but just take a breather. I'm not going to even pause the camera or anything. So. And I'll post these videos like after class so that you guys can hear me dragging on again. What are you done? You've done this enough for the
That's all of this right now, but
I'm gonna paint my face. What's that?
I'm taking two online courses at the senior center. Are you doing anything else?
Yeah, genetics and theory. Theory and practice.
That's right.
I'm glad you're in here. You're into genetics?
Yes, that's odd. I would like something I should be able to.
Who did you have genetics for?
As excellent as many times. I know how to write it.
My genetics class is
Yeah.
not.
That was me too.
Scientists,
medication and stuff.
What? Well,
Just say you know how to write.
my genetics class is a little harder than this, but not much.
I'm taking genetics right now. So,
You're not taking it with me though.
no.
I don't know
I'm
how our
taking
class
lab with you.
is.
You're not teaching lecture.
No, I'm not teaching.
I just wish it was taught in person because I hate online.
Yeah, I think for a class like that too, it's more helpful, but. All right, guys, let's get back to it. I want to go through this next section a little quickly. Again, we've got so much stuff to cover. So the third section in this chapter, I took it out completely. It's all on microscopy and methods and things like that. We're going to get into some molecular methods in the lecture, but a lot of that stuff is going to spill over to the lab. So I took that section out completely. So I wouldn't even bother reading 1.3 unless you are like super nerd, and that's okay. But I got enough nerdy stuff for you up here, I think, as it is. So, all right, that's not going to be on your test. A lot of those things we're going to talk about in the lab, they'll be on that lab midterm and final, but don't worry about it. So I did include this though, because moving forward, we're going to bring up these organisms a lot, but this isn't anything crazy difficult. Just looking at, when you want to study biology, right, putting a bunch of people in a blender and grinding them up to study their protein mass is not something that's very feasible, but it's something that you could do with flies or worms, things like that, or little zebrafish. So what makes a good model organism, right? These are some features that all model organisms share. So this is all you need to know in this section right here, right? Because moving forward, it's just going to say like, well, the fish has really good genetic tractability and so on. All right. So genetic tractability, that means that you understand the genome, right? So maybe it's been sequenced. That's great. So you have sequence information about the genome, how the genes are divided up, what the genes do. The ability to genetically modify the organism is very important to make genetic mutants and also have mutants available that have already been established and characterized by other scientists is important as well. C. elegans is a great example of that. It has a very close-knit community within the scientific community of individuals that have banked different strains of worms that they share, at a very low cost between each other. A horse would not make a very good model organism, right? You're going to need a big barn and a lot of food, and it's got a very slow reproductive time, and you're only going to get maybe at most 2 twin horses, something like that. You want an organism that has a short generation time and has high fecundity, right? So fecundity means it produces a lot of organisms. When a C. elegans, a worm, drops eggs on a plate, it drops upwards of about 100 to 120 eggs. That's, one reproductive cycle, and now you have a whole bunch of worms. All right, so horse, not a good example because of ease and cost, right? It's going to be very expensive to maintain, takes up a lot of space, a lot of food. You want an organism that has conserved biological pathways, right? So if the biological pathways of what you're studying is completely different from a human, if you're trying to study medicine or something like that, it's completely useless. So when we say conserved means across species. right? And we say biological pathway, we don't mean just signaling pathways, right? So how molecules work with different molecules to ultimately have some endpoint, but also metabolic pathways, all right? Enzymatic pathways, to process energy and whatnot. And it's really helpful if somebody's already been studying it and there's a lot of information about it and you can build on that information. Okay. So what are good examples of model organisms? E. coli, definitely fundamental to cell and molecular biology because of all the ability to study the molecules in E. coli, right? So, studying DNA, ultimately very important. And in order to study this string of hundreds of thousands of letters, you need to be able to cut the thing up into smaller pieces. And there are molecular scissors, enzymes that are found in bacteria that do that. They will cut DNA in predictable manners at specific sequences called restriction enzymes. And that's almost your first step into breaking it down into a manageable size that you can study and now analyze the sequence. Oftentimes you need to increase the amount of DNA. Like if you're trying to study one gene, just having one copy of it sitting in a tube is not going to work. You need a sizable amount of DNA to be able to, for it to be able to work with the equipment that you have, like a DNA sequencer or an agarose gel. So E. coli present a way to bulk up that DNA, to copy the DNA for you using its replication machinery. And E. coli can also produce protein for us, like insulin is produced by bacteria. We say it's a recombinant protein because it's like the natural insulin gene, but it's been modified a little bit genetically. So maybe it's had a tag stuck onto it or something like that. And that's when we talk about, if you guys watch those fancy Pfizer commercials and you see rDNA, that's what that means, recombinant DNA or recombinant protein. Any recombinant molecule is a modified molecule, the genetic level and the protein level. All right. These E. coli can act as machines to bulk up that DNA and bulk up proteins so we can actually analyze it on a machine in the lab. They grow in colonies, and what's nice is, so if I pick one of those colonies and I analyze all the cells in that colony, it's a guarantee that they are all clonal. They're all genetically identical, physically identical, they're completely identical within one colony. On the same plate with the same bacteria, chances are all the clones of all the colonies are the same, but you never know. It could have diverged out. You might have had contamination. But the fact that you can grow these on an agar dish makes it very easily save space. It's low cost. And within one of those clonal populations, you have millions of cells, right? Millions of bacteria can sit on the head of a pencil. And it's very tiny, but contains a lot of organisms in there to study. But E. coli are prokaryotic, right? So they're a great tool in molecular biology. We use them all the time to do our experiments, but we're not necessarily studying them unless you're a microbiologist. We want to study eukaryotic cells, for the most part. And so yeast is an example of a eukaryote that's single cellular, grows abundantly, but it has all of those eukaryotic characteristics that are important to us, such as the central dogma, transcription and translation. And it's a eukaryotic system because the prokaryotic transcription and translation system is significantly different. Also, it contains what's called, or has the ability to do post-translational modifications, something that bacteria can't do. So after the DNA is read and RNA is made, and the RNA is read and protein is made, there are important post-translational modifications that occur to that protein. And eukaryotes will do them. So yeast is an example of a cell type that'll do those, so you can study those in yeast. Right? All these things, DNA replication, transcription, and RNA processing, the cell cycle, all gave rise to other fields like cancer biology. And they were all discovered initially in the yeast because it's a simple eukaryotic organism that we can use to study these processes. There's a yeast, when it divides, it doesn't, its manner is a little different than the typical eukaryotic cell that we do. that we study, but it buds. But it still has similar cell cycle components, cyclins and CDKs and all those things we're going to talk about later on. Now we're getting into multicellular organisms, and the worm and the fly are a great multicellular model organism. They're cheap. They have high fecundity. And now we're actually dealing with a real organism. And you can take a bunch of worms and put them in a tube and grind them up. You can analyze all of their chemical components. C. elegans are great because every adult worm is going to have exactly 959 cells, and we know exactly where every single cell derived from. Over from starting at a zygote, when that split into two, the pathways that those cells developed into, we know every step of the way that took in the worm. So developmental biology basically came rise as a result of studying C. elegans. C. elegans are easy to genetically manipulate. You can actually just feed them bacteria to genetically manipulate them. We'll talk about that later on. You can also inject DNA into their gonads if you need to make a stable type of genetic variant. And same thing with the flies. You can also do microinjection to genetically manipulate them. Now, Drosophila is a key organism in the study of genetics as well, because it has a lot of similarities to how our X and Y chromosomes interact and work, but also in developmental biology as well. Have you ever seen pictures of the flies with like arms growing out of their eyes and stuff? A lot of that is because of manipulations that you can do with developmental biology. The worm has a real simple anatomy. It has this really thick cuticle that can make it difficult to work with sometimes, and it evolves in different stages. It has a life cycle that starts out at larval stages, and then it goes into a full-grown adult. And the fly, anyone here taking genetics? Some of you guys are. So we're going to use the fly. We're going to use flies and worms in genetics. I don't think we're doing any C. elegans in cell bio. We're focusing more on proteins and stuff in cell bio. But you guys are going to do crosses in these in genetics and how the fly things. All right. So again, I'm not a plant guy, but I never worked with Arabidopsis. But photosynthesis was basically discovered, the molecular process of photosynthesis was discovered at Arabidopsis. And, these individuals that do, I've seen Arabidopsis, I call them farms, but there's a better term for it. I can't think of it off the top of my head. But these things grow like weeds, and they're really quick. They produce thousands of seeds, and still widely used by botanists today to further study photosynthesis and cell growth, responses to the environment. All right, it's a small plant, easy to grow, high fecundity. The zebrafish, great organism because it's highly transparent. And so that means that you can see all the tissues and stuff under the microscope. And it's a vertebrate, right? So now we've got an organism that's really getting a lot closer to humans. And so if you're trying to genetically manipulate the zebrafish, it's relatively easy because the embryos are very clear. They develop very quickly, right? Within a few hours after fertilization, you've already got presence of major organ systems. We know a lot about the genetics of zebrafish. And like I said, they're vertebrates. They have a spinal column. So there's a lot of genes that are homologous to humans. There's a lot of conservation in terms of those molecular pathways. And these labs are kind of cool. They have these great aquatic setups with all these fish tanks. And the mouse, now we're getting into mammals, and there's a lot of homology to humans. Almost 85% of the genes found in a mouse are homologous with humans. They have physiology, they have organ systems and tissues. You can start to study disease models in mice. This is an excellent model in studying diseases like cancer and Alzheimer's and Parkinson's. There's a lot of information about the mouse as well, genetically available. There's different genetic strains available commercially. They're a little more expensive and a little more work to house than your worms and your dishes. And obviously, there's issues with animal rights even when you get to this level of model organism, but extensive information available for the mouse. I don't know why the author made this picture. I think that's a little weird, but that kid have earrings already? And they should put earrings on the mouth. I think that's. All right. So today, though, when you're doing cell biology, the majority of cell biology, you just need cells to study it. You don't need these model organisms to do it. And animal cell culture is a great way to do that. Now we can actually grow cells in the lab with relative ease at relatively low cost. And we can study those cells. We can put them in a solution and study how they behave while they're living. We can kill them and break them up and study the chemicals and size. We can visualize them under the microscope. So animal cell culture is a great tool. Basically, if I took a student up here, I cut them open, and I took a chunk of liver out of you, I could grind it up and stick it in a growth medium, put it in a carbon dioxide incubator, and the next day, some of those cells will stick to the dish. And when they adhere to the dish, that's a good indication that they're still viable. A lot of the cells are going to be floating around in the media and they're all dead. We could dump the medium off, just now start to grow these primary cells, which they're called. As they divide, they will start to cover the surface of the dish. And those will grow for a couple weeks and then they'll just crap out. That's how it works. However, there are ways that you can infect them with different viruses to make them immortal, right? Also, if you happen to have a tumor in your liver and we grab some of those cells, those would divide continuously over multiple generations. So these immortalized cells become more useful because we can continually grow them in the lab. We don't have to keep killing an organism to take its tissue out. And there are thousands of immortalized cells available to study, right? The big company that distributes these is called ATCC, American Tissue Cell Collection. And, they're like a few $100, and as long as you have a sterile environment to grow them in, you can grow them abundantly. All right. Probably the most famous cell line is the Helix cell line, which came from an individual, Henrietta Lacks. And I think these are probably Helix cells right here. So I'm also teaching a cell culture course this semester. I did this for years at Moffitt and my lab at USF, and really good model to do a lot of different things. All right, viruses also, this isn't a virology course, but we use viruses as molecular tools. We use them to infect cells with certain genes. Right? That's one thing about a virus, right? It's a non-living entity that can basically inject its nucleic acid into a target cell, and that nucleic acid will cause the cell to hijack, like hijacks the machinery of that cell to copy the virus. High replication rates, right, has medical relevance, not only in terms of virology and viruses that are dangerous, but also how our immune system responds to viral infections. Viruses are simple, non-living, acellular things because they don't have the ability to do metabolism. They don't have the ability, importantly, to divide on their own, right? So they're considered non-living. All they are is a coat of protein that's surrounding a specific type of nucleic acid. Whether it's DNA or RNA indicates a big area of divergence in virology. You have DNA viruses and RNA viruses. But that's really it. It's just nucleic acid with a protein coat, and then some viruses might have a capsid which surrounds it. Also proteins that might come out, like the SARS virus has these spike or attachment proteins that come out of it. Okay, so we call them particles, not cells, because they don't have a membrane that surrounds them. All right. I would just take a breath, so break. What time is this class over? I'm gonna be asking you that all semester. Okay, and everyone knows except me. All right. So this is not chemistry, okay, and I understand that. And we're not gonna get crazy. I'm not gonna see a lot of equations on your exams or not, but we need to get all to the same level if it's been 20 years since you've taken chem or you flunked out of it. With just some basic principles within chemistry and physics, just to kind of understand things moving forward. This is where, though, the volume of information kind of gets up a little tick. So, so this is, we're not really concerned too much with inorganic chemistry, obviously, because living systems are made of organic molecules, right, that contain carbon, oxygen, hydrogen. And we're more concerned about how the chemicals interact with each other, chemical reactions, to enable things like polymerization, right? The building of larger molecules, such as longer nucleic acids or longer proteins. And also importantly, how energy is metabolized, right? So how ATP is made, how it's used, what it does. In order to understand that, you have to understand a little bit about just some basic principles of thermodynamics. We're going to get into delta G a little bit, but not too crazy. So how atoms interact with each other is through molecular bonds, right? You've got different bonds, covalent, ionic, hydrogen. And what makes them different, big importance to us, is the strength of those bonds. Covalent bonds are considered like a permanent bond between two atoms. Can be broken enzymatically, but it would take a very low delta G to do that. The most diverse type of bonding situation relates to the atom carbon, right? Because carbon has 4 electrons in its outermost shell. It can form up to 4 bonds. So now you can produce these very elaborate carbon skeletons, which makes carbon the ideal atom to build organic molecules with. Remember, life needs diversity and complexity. So when you look at these atomic models of these different carbon-based molecules, so like simple methane, CH4, has 4 hydrogens that are equally distributed around the carbon. And that equal distribution also means that all of the electrons are equally distributed around the molecule, right? So imagine those things are spinning around, the hydrogens spinning the carbon, all crazy like, and if you take a picture of it, in general, they're all equally distributed. Remember, the electron carries the negative charge, right? The proton within the nucleus contains the positive charge. And if there's equal distribution, that means the charge is equally distributed. And that means that the molecule is nonpolar. And here we get into some important terms that are going to be used over and over again throughout our study of cell bio. So what does nonpolar mean? Well, in general, it generally means it's relatively non-reactive because it has an equal distribution of charge. As we're going to see in the next bonding example, ionic bonding, it's charge distribution and how electrons are situated, which produces that bond. But a molecule like water, for example, you have an unequal distribution of electrons. Oxygen, which is much more electronegative than hydrogen, pulls the electrons on that side of the molecule. So all those electrons are spinning around that whole thing, but if you took a picture of it, like a freeze frame, you would see that there are more electrons over here. Having more electrons over here makes this side of the molecule negative and makes this side of the molecule positive. So now you have a difference in charge. And because of that, oxygens can start to bind up. Water molecules can start to bind up. Okay, so a negative charge here because electrons are being pulled on this end, and a neighboring water molecule, you'll get an interaction between that negative and that positive charge, and we call that hydrogen bonding, which is another type of bonding. It's that hydrogen bonding which gives all of the unique properties to water. What's some weirdness about water? What makes it a really great, like the stuff of life? What's that? Yeah, it's cohesive, right? Because of the hydrogen bonding, right? So, you know, a tree, how does a tree get water? Is there like a pump in the roots or something? Pumping it up from the ground? It's actually caused from evaporation from the leaves, right? And that cohesiveness of water pulls the water up from the ground through the tree. Water is also a great solvent, right? A lot of things can dissolve into it because of its polar nature. It can do evaporative cooling, right? Can harness heat very well. It also expands when it freezes, which is odd, right? That makes it more buoyant, float on a surface. Very important for the planet that the glaciers are floating and reflecting a lot of sunlight. But so these covalent bonds can be divided up into two different groups: non-polar covalent, where you have the equal distribution, and then polar covalent, where you have that electronegativity pulling electrons on one side of the molecule and creating two different charges across the molecule, or a change in charge across the molecule. All right, so these things now start to, you can start to look at chemical reactions, right? So for biologists, the two big ones are dehydration synthesis and hydrolysis. Okay. In dehydration synthesis, you are building molecules. You're making them longer, you're polymerizing them. So we have a chain of amino acids here, and we have amino acids sitting out here. Through dehydration synthesis, we can create a covalent bond, add a new subunit onto that growing polymer. Now, it's called dehydration synthesis because in order to open up and free up electrons, it actually has to lose some water to do that. Right? The opposite of dehydration synthesis, if we're not building, then we're breaking. And hydrolysis is the opposite of dehydration synthesis. So instead of losing water, in order to break this bond, we have to bring in a molecule of water to soak up those free electrons and make the whole reaction stable. Right? In these reactions, you have reactants, the starting materials, and then finally products, right? So is this dehydration synthesis, or is this hydrolysis that's occurring from left to right? Right. And we're doing hydrolysis from right to left. All right, so covalent bondings are strong, almost considered permanent bonds. Ionic bonds, though, are a little different, and they work on the ability for certain inorganic chemicals to form ions in solution. Okay, so an ion is an atom that is charged, and it's charged because there's an unequal number of electrons compared to protons. Right? If you have 17 electrons and 16 protons, you're going to have a negative net charge. And when you have ions, or when you have atoms that associate together, oftentimes they will move their electrons from one atom to another. The simplest example is what happens with sodium and chlorine. So the sodium atom has 11 protons and 11 electrons, so it's electrically neutral. The chlorine atom has 17 protons and 17 electrons, so it's electrically neutral. However, if you remember from chemistry, the valence shell, which is the outermost shell of the atom, wants how many electrons? Wants 8, right? So when it gets really close to having 8, it becomes very reactive. And in the case of sodium, it only has to lose that 1 electron from its outermost shell, and now it's stable and has 8. In the case of chlorine, it only has to gain 1 electron in its outermost shell, and it's now stable because it has 8. So when these two come together, they're not dumb. Sodium knows it donates its electron from sodium to chlorine. They're much happier. But that donation causes a change in charge. You're giving that atom a negative charge. So chlorine now becomes Cl minus, and sodium now becomes Na plus. That opposite charge is enough to create what's called an ionic bond, right? Similar to the hydrogen bonding, which we talked about. But this actually involves a donation of an electron, and that donation is permanent. When those things disassociate, they retain those charges. That's what we call like an electrolyte, right? It comes apart in solution. So yeah, you can see, and because of the polar nature of water, right, these ions will interact with water in a certain way. The positive charges will interact with the negative charge of water, of oxygen, and the negative charges will interact with the positive charges of hydrogen. That's how they'll orient themselves when dissolved in solution. And they will also form these lattices that grow, these crystal lattices, because of the charge separation as well. Okay, but water ultimately very, very important to understand that it's polar and to understand that molecules that are also polar will kind of interact with water in a weird way because water has the ability to hydrogen bond. The hydrogen bond is a weaker bond than the ionic bond, but it's very, very important in biology because it's very dynamic. An example that I talked about already today is like a double-stranded molecule of nucleic acid, right? And those A's and T's and C's and G's are held together by hydrogen bonds. That's what holds the two strands together. And there is an enzyme called DNA helicase that can go on and cut that thing apart. So if anyone ever asks you, you could be any enzyme in the world, what would you be? You say DNA helicase because you can unzip their genes, right? It's about as funny as it's going to get in this class. All right, so hydrogen bonding, really important. It also helps provide, in an aqueous environment, these protein molecules, how they're going to fold up, right, is dictated a lot by the polarity of its environment. You can see, you have this thing that if it was like an inorganic solvent, like an ether or something, that's highly nonpolar, it would not fold up into the correct conformation because you couldn't do this hydrogen bond. All right. It also permits us to have that hydrophobicity and hydrophilicity that we talked about with lipids, right? So, nonpolar molecules are considered hydrophobic. Polar molecules are considered hydrophilic, which means they're happy around water. They will dissolve in the water easily. Whereas nonpolar hydrophobic molecules such as fatty acid chains that make up the triglycerides and phospholipids, they don't dissolve in water. That's why they're good structures for membrane. All right, why don't we take 5 here and before we keep going and
Like,
But thinking about it, then
it's, oh, yeah. It's not that the only, it's not,
it's one of those things, you know. I took more from this. We didn't use a lot.
They don't pay attention to you, I'm worried about what person wants. I have to go with this.
Genetics, she.
Oh,
But for biodynamic, it's big and she was a home. She was home. She actually do questions. She actually do questions in the, so if you don't understand her, first, she's gonna talk for the time, okay, I'll give you a I didn't bring my charges, I didn't bring the earpiece of my charger. And play the quarter. The box. I'm a.
There you
So
go.
I had a box.
Up.
For work, but if the teacher. Yeah. Because I have to get my, it's my safe. How I use it for phones. Yeah. This is a. So much, that we have to get the next. It's a problem. My quicker, that's why I like, I would use it. It's not the better option, y'all. If you might not see it, so I'm not going to, not. I was looking most of the things, right? Yeah, yeah. Which is why, I have a small little one that I do my papers and stuff, because that the time, that was next session. So.
Take the lunch.
Yes.
yeah.
Just so I don't get overloaded.
No.
How.
So that could
One of the stuff. One of the part. Word doctors. I mean.
Now, most of the time, because if you don't, you turn it into zero to their. And especially like using the H, they can't open it.
So you can download the word app. And like all the.
So that's what I have, there's a PowerPoint, Word, PowerPoint, that Word, and that comes out on my phone. Because that way when I do it was shown here and I can download it as a file. But I'll let it.
Also, you have to get money.
Yeah, the Steve, let's see, after this. Yeah, thanks for not.
It's not as good.
No, it's like. You can't lose it for nothing. You can't, do anything anymore.
It's mostly your client.
Yeah.
It's just.
Oh.
This is right.
That's what I should say.
It's just God.
I
I don't want to dog really.
That's fun.
Yeah. Yeah. So if one says. So pass.
Are you?
That's the amount, but you also need to sign.
It's not bad, it's not much so bad, especially with the Cambridge, it's an animated Cambridge.
I just finished.
This one.
Yeah, we're going to.
Yeah, it's just.
Computer is very. I passed my first.
Did you say, Dr. Musgrave?
You know what, helped me pass him is memorization.
Well, see, I took the summer. Now.
It's a little in the summer.
That's why you did
Two.
Yeah, we scored about two in the summer. And we left, I mean, my.
But the trade that Dr. Musgrave did, if the tickets that it was making that.
That was my sense. Figure out.
I didn't do too, I mean, like I said, I passed my good grades.
When I felt for the first time, when I felt we're going on the first time, I took it, like, too bad your personality don't get you pregnant.
Like a.
So you can do money from my house, so I think it's right.
Yeah, and he's understand, he's stony, but he's not.
Yeah, I was gonna say, my friend, she told me, she was also, yeah, he's kind of like rude, and I was like, well, not.
He can be, but you have to butter him up.
Like every time I emailed him, he was never really rude to, so I was like, okay, we locked in. I'm really trying.
But if you butter him up. You have to butter him up.
Right back.
And then when I was like, I don't work, I built the first, I'm sleeping again, I said, so those are my hands, so I still, he's from the end of the lane. But I'm like, hey, Chad, I thought we were going to strip the method. And I was like, are we not? He was like, I'm going to do something. And then I was having difficulties with my mom, like, I actually stayed here in one of the days. So then we had my, just about that. We started for yeah, my birthday. We didn't bring inside her to the store for, you know, I was and he was the better cook. And then we got his name was like. And she said she lived in and I never got into such a big reason that they could have. Really understand what he's saying. Most things. Something a lot of fingers, but it didn't give me. So like I just went, my friends now. Give me a whole lecture. Is that a? What is this? You don't take it down.
Theirs is that, you know, they don't see you in person, it was very hard to get that expression. Email them, that's what it is, about grabbing and trying to get up and saying, you're. I knew she knew my name, I came back, I don't see the people around her, and where I was at, one of my.
He don't think he's great, but still like, but it comes to drawing things again.
See, that's why I would get her online, we didn't have to draw, we did this to the. For the readings.
What about my shoes?
Yeah, I'm. Oh, for labs. So, what we had, our teacher, felt like we find most of the reaction all the time. I don't want to.
All right, guys, let's keep plugging away here. All right, so now why are we talking about this? Thermodynamics. Doesn't sound very biological, does it? Thermodynamics is more physics. But all we're concerned about are chemical reactions, and if the chemical reaction is something that's favorable or not. Okay? Because if it's not favorable, then life has to give it a little push to make it occur. Right? It's how you get all that diversity in terms of molecular structure. The laws of thermodynamics can kind of explain how reactions can be unfavorable or favorable. And if they're unfavorable, what we need to do to overcome that and make the reaction proceed. Okay. I get into here how Gibbs free energy is derived, but ultimately all you really need to understand is Gibbs free energy. Gibbs free energy is that delta G that hopefully you guys remember from Gen Bio. And the value of delta G indicates to us if a reaction is favorable or not. Right? If it's a positive number, right, it is unfavorable. So most reactions that go on in the cell are unfavorable, which means they require another reaction to come along and couple to it to assist it. If the reaction is less than zero, it is energetically favorable, which means it does occur spontaneously. It'll occur in solution. It just occurs on its own. It doesn't need an enzyme or anything like that to assist it. So, and if it's zero, that means it's equilibrium, right? It's in equilibrium. The number of reactants and products is the same. The reaction is going back and forth in the same manner. Most reactions that we're going to study, especially here at the beginning, all of them, are going to have large delta G values that we need to explain how you get that number down to make the reaction overall favorable. But it's derived from these three principles or so and two thermodynamic laws. The first law of thermodynamics states that you can't ever create or destroy energy. Right? You can only transfer it or transform it, move it from one system to another. Now, this is important in biology because cells need to take in nutrients from the environment that they break down and extract that energy from the hydrolysis of those bonds. You can take that energy and then transfer it to another molecule like the ATP molecule we talked about, which is the currency of the cell. And then you can use that ATP molecule to get energy from, right? The second law of thermodynamics has to do with entropy, and this is disorder and order in the universe and how that applies to biology. So biological systems, look at the sentence here. So living systems increase local order, but overall increase entropy through energy release, right? So the universe is always moving in a direction of disorder. A good example I like of this is you build a brick wall. Right? So you're going to put energy into that to build up that brick wall, right? But over time, that wall is going to fall, right? Over time, it's moving into a state of disorder. That wall is going to come apart. And in that process, you get a release of energy, right? The bricks are falling, gravity's pulling it to the earth. You could capture that and transfer that energy to something else. Okay, that's the second law of thermodynamics. It all relates to how heat is moving in the system, and that's what enthalpy is, right? You have the release of energy, which is exothermia, and you have the absorption of energy, which is endothermia. This is an important factor in deciding if a reaction is favorable or unfavorable. So all these three things kind of come together, and it's derived in this equation, delta G equals delta H minus temperature. So temp is a factor here, times delta S, right? So to come up with your delta G value, which is ultimately what we care about. Again, we want a delta G that is greater than zero, but we rarely get it. In order to get a delta G that is below zero up into a positive number, we have to couple the reaction to another chemical reaction that has usually a very high delta G value. Okay, so here's an example, and this sort of stuff is as complex as we're going to make it with terms of like calculation and stuff like that. All right, so the reaction of going from A to B, right, has a delta G of plus 10 kilocalories per mole, and that's the unit for Gibbs free energy. It's a measure of energy per mass. All right, so if it's plus 10, is it going to go forward spontaneously or not? It's not, right? So we need to couple it, all right? And when you couple reactions, basically they're occurring side by side, temporally and spatially, and they're going to interact in some sort of manner, right? Atoms and functional groups are going to transfer between different reactants and products. So we couple it with a different reaction, like going between C and D, that has a very low delta G, a reaction that does readily occur spontaneously. And all you do is, minus 20 plus 10 gives now a delta G that is below zero. So the reaction is favorable and it will proceed. Okay. So an example like this might be some sort of dehydration synthesis reaction in order to build a polymer of nucleic acid, in order to increase the size of a DNA molecule. That is an unfavorable reaction. So we have to couple that with enzymes and the use of ATP to lower that delta G, right? All right, so, and this is, ATP is a great molecule to transfer energy because, and it's called the energy currency of the cell, because it's rapidly being used and rapidly being produced because it has these triple phosphate groups, 3 phosphate bonds, and the phosphate bonds contain a lot of energy in them. ATP can also exist between these different conformations. You can have adenosine triphosphate, adenosine diphosphate, which is 2 phosphates, and adenosine monophosphate, which is 1 phosphate group. And they can build up back to triphosphate and break down back to adenosine monophosphate. Typical ATP hydrolysis, right, which is the removal of just 1 phosphate group, has a relatively low delta G of -12. So it will occur spontaneously, and it will occur very readily. And that delta G can be coupled to other reactions that have a very high delta G to bring that down. Right, so here's ATP going through its phosphorylation and dephosphorylation event. Okay, you can see ATP, which is made up of a ribose sugar. It also has adenine attached to it. Adenine is related to nucleic acids, right? AT, A is the adenine. And it has the triple phosphate, okay? So through a hydrolysis reaction, which means water's coming in, right, you are freeing up, you're taking up those free electrons that occur when the phosphate breaks off, and now you have a free phosphate that's available, okay? The process of breaking that phosphate-phosphate bond releases a lot of free energy. That energy can be coupled to the other reaction. It's just immediately absorbed into the reaction. Now, what's great about using ATP is not only do you release energy, but you also release that phosphate. And oftentimes, in many reactions, that phosphate will attach onto something to modify it chemically. We'll see that going forward. The opposite of hydrolysis is dehydration synthesis, right? So that's going to be, 2 phosphates, another phosphate coming in to produce either ATP or ADP. This figure isn't showing the dehydration synthesis, it's showing hydrolysis of either 1 or 2 phosphates. But that ADP or that AMP can recycle back to a double or triple phosphate state through a dehydration reaction. Now, we call these things something else as well. We'll call it, for specifically ATP, we'll call the hydrolysis event just dephosphorylation. Typically see that. And we'll call the dehydration synthesis event phosphorylation. So, when you're moving phosphates, you're also moving energy or moving energy as well. Okay. That's it for chemistry, right? Bonding, chemical reactions, delta G, coupling reactions to make reactions more energetically favorable. Now, in the next section of the chapter, we're going to get into the macromolecules, which are large molecules, right? Now you can't just have life based on these molecules made up of 1 or 2 atoms. You have to have complex molecules that are big and can take on a diversity of shape, because structure determines function. So here we're going to go over the macromolecules. What are they? The big molecules that make up cells. There's only 4 of them. Well, nucleic acids, proteins, lipids, carbohydrates. Very good. Yeah, those are the 4 main groups. It's kind of nice. There's only 4 of them, but there's a lot to them. But they make up about 90% of the dry weight cells. It's a lot. All right, first one we're going to talk about are the carbs. So carbohydrates, what are their purpose? Well, they're a great source of energy. Carbon-carbon bonds are high energy bonds. So if you break them apart, you can couple that energy release to other reactions, such as the production of ATP or the transferring of electrons. They're just made up of carbon, hydrogen, and oxygen. Okay. But they also have structural roles as well. So remember, we were talking a lot about those post-translational modifications, modifications to proteins that occur following translation. Glycosylation is a common post-translational modification. These carbohydrate groups can add on to proteins to modify the protein. Carbohydrates can also embed themselves in the plasma membrane, sticking out of the membrane. An important component of the immune system is tolerance, right? The immune system can tolerate the cells in the body, right? If it doesn't tolerate it, that means you're having like an exaggerated immune response against those molecules, and that's not good. The way that immune cells identify cells of belonging in the body is through carbohydrate molecules embedded in the membrane of the surface. You guys remember back from like in MHC1 and MHC2, all that stuff related to carbohydrates. They're divided up into a few different groups. Monosaccharides means one sugar group. Oligosaccharides, you have a few sugar groups. There's another important group that's really not mentioned up here called disaccharides, because there's a lot of important disaccharides in biology, but you're okay. And then, of course, polysaccharides. These are the larger, right? So the larger you get, the bigger your polymer is, the more energy you have, because the more bonds you have. But also the more difficult it is to break that carbohydrate down, right? So there's a really good principle here, the Atkins diet, right? So what does the Atkins diet do? It starves your body of carbohydrates. And that's because the preferred carbohydrate of the cell to extract energy from is the glucose molecule. So the cell can have everything around it: proteins, lipids, triglycerides, glycogen, but where it's going to go to first is glucose. And it's going to keep going to glucose until glucose is not available. And then it's going to go on to start breaking down lipids and things like that. So if you starve the body of carbohydrates, including glucose, ultimately your cells will resort to breaking down larger molecules like fats, and you'll lose weight, right? Or you can just take Ozempic. All right. So, all right. So how do these carbohydrates get formed? Well, through the process of polymerization, and the bond that forms that, or permits polymerization, is called a glycosidic bond. Right? A glycosidic bond, the formation of that is unfavorable. So in order to do that reaction, we have to couple it with a favorable reaction. So let's look at, well, not this slide, but the next one, I think. So this is the simplest monosaccharide, right? Glucose. So glucose can exist in two forms, either linear or circular. When it's dehydrated, it's in the linear form, but when you throw it in solution, it rings up. Okay? C6H12O6. All right, you should know that. And you should be able to, be able to readily recognize a glucose molecule, maybe even draw it. But it can exist in two different isomeric forms, right? Alpha and beta, and where just the rotation of that bond is different, right? You have the hydroxyl functional group and coming off on that side there, and on that side in the alpha form. So this is important because different complex carbohydrates are made of maybe the different form of glucose. But this is the preferential molecule to go to that the cell gets energy from, because it's the starting point of glycolysis, right? What we talked about earlier, remember, glycolysis to photosynthesis to oxidative phosphorylation. And this is where ATP ultimately mostly comes from. All right, but the formation of this glycosidic bond, I know this is kind of a small figure here, but so figure A of there is just showing you the bond. There's the alpha 1,4 glycosidic bond. And then B here is showing you the pathway that takes place in order to create that bond. But remember, formation of that bond has what? Very high or low delta G. It's high or low? Right, yeah, it's energetically unfavorable. Okay, so we need to couple another reaction to it to, change that delta G to make it favorable. So there's a few things that occur. One is we're going to use ATP, right? So we take glucose. Remember, we're trying to stick two glucoses together, right, to form a glycosidic bond. So we take some ATP, energy gets released, and it puts in, and a phosphate gets transferred to glucose to make a chemical intermediate, which is G6P or glucose 6-phosphate. Okay. Glucose 6-phosphate then changes to glucose 1-phosphate, right? Because you can see it gets transferred to another side of the molecule. And then this is the most important step here through this addition of a chemical called UTP, right? You get an intermediate molecule that gets made, which is called UDP glucose, right? Uridine diphosphate is the intermediate form. This is a higher energy form of glucose. Okay, you can see it's got these additional phosphate groups on it. Now this can lose the UDP, and in the process of losing its UDP, enough energy is available to stick two glucoses together. Okay, so you have, the actual coupling to provide the energy to form the bond is provided by having this energetically favorable UDP glucose. The process of getting there, though, involves the use of ATP. Okay, so a good example. You're going to see this as we go through pretty much all the macromolecules. Doing dehydration synthesis is a process that is energetically unfavorable, and there needs to be some way to overcome that. And with carbohydrates, it's the formation of this higher order intermediate called UDP glucose. All right, polysaccharides get a lot bigger and a lot more complex, and that's why we call them complex carbohydrates. Different forms of them have different the different alpha or beta types of bonds that make them up. And the more complex you get, really the harder it is to digest, right? So we can't break down cellulose. It's just becomes dietary fiber. For organisms to be able to break that molecule down, they have to have specialized enzymes to do that. Glycogen is an important sugar group because it's what animals use to kind of store carbohydrates in the body, right? So glucose will basically get bonded together in the liver to produce glycogen, and then glycogen can be stored in the liver or stored in muscle cells. And if the body is ever depleted of glucose, it can start to break down that glycogen and undergo glycolysis. Plants don't store glycogen, they store cellulose instead, and they use the cellulose to build the body of the plant. All right, that's it for carbohydrates. Lipids are a very, very good energy storage molecule. In fact, per mass weight of lipid, you have more energy in a lipid molecule than you do in a carbohydrate. However, they're more difficult to break down, largely because they're mostly hydrophobic, and it's hard to get enzymes to work with lipids. You have to do another special process to break them down. That's why glucose is the preferred molecule to get energy from. But the fact that these lipids are made up of these long hydrocarbon chains that contain dozens of carbon-carbon bonds provides them with a lot of energy. Right, so we call these fatty acids, right? They have a carboxyl group at one end, and the fatty acids can exist in one or two conformations. We can either say the chain is saturated, which means it has the maximum number of hydrogens associated with all the carbons, or it can be unsaturated, which means it's missing a couple hydrogens, and because of that, it has a double bond present. Right, so here's three different fatty acids: palmitate, stearate, and oleate. And these two are saturated here. The easiest way to recognize it is there's no double bond. But oleate is an example of an unsaturated fatty acid chain because it doesn't have the maximum number of hydrogens. You're missing a hydrogen here and here. And because of that, the electrons take up or produce another carbon-carbon bond or a double bond. Okay. And that is important structurally for the lipid as well. So the formation of a double bond actually produces like an outward kink in the molecule. And the change in that structure actually allows it to interact differently with other lipids. Saturated fatty acid chains, because they lack that kink, can actually snug and fit a lot more closely together. It makes them more sticky and it makes them aggregate more. And it's saturated fats that build up in the blood vessels and produce atherosclerotic plaque, right? Gives way to atherosclerosis. You know, the unsaturated they're bad too, but they're not as sticky as the saturated fats. All right, the most fundamental lipid molecule is the triglyceride, right? And it's actually got 3 of those fatty acid chains that are stuck together with a glycerol moiety, right? A group on the molecule. Glycerol is like a lubricant, it's a slippery molecule. And along with that very non-polar fatty acid chain, the entire molecule is very hydrophobic. Okay, we've got a lot of non-polar stuff here, and we've got that glycerol stuff, which is not highly polar. Okay, is your glycerol right there. All right, typical fatty acids. This is like when you're cutting the fat off a steak, that's what this is, triglycerides. Okay. All right, continuing on with lipids. Now, if we take a triglyceride and we remove one of the fatty acid chains, now we've freed up some electrons. We still have that glycerol, and we add some phosphate. Now we're adding a very polar group, the phosphate, to the head of the molecule. Okay, and that's what makes that head hydrophilic, right? The polarity of the phosphate can interact with the, with the water molecules around it. And this makes it a phospholipid, right? Because it has the polar head that is hydrophilic and the non-polar tail, which is hydrophobic, we say these molecules are amphipathic, right? They have differing physical chemical characteristics on opposite sides of the phospholipid. All right, now in GenBio, you probably didn't talk about all the different phospholipids that make up cell membranes. It's not just one. It's a family of them, and lipid biologists study these in extreme detail. But they all have similar shapes. So the most simple is the phosphatidic acid, right? This lacks any extreme functional groups that are attached to the phosphate. You can see they all have a glycerol moiety except sphingomyelin. Sphingomyelin is the one exception that doesn't have glycerol. Instead, it has an amino acid that gets stuck in there to produce this phospholipid. Phosphatidylanolamine, phosphatidylcholine, phosphatidylserine, and phosphatidylinositol, all of these have additional functional groups that are attached to the phosphate groups. Okay. Inositol is a type of sugar. Choline and serine are related to amino acids. So how do I know serine is amino acid, but how do I know that like choline is an amino acid and serine and ethylamine are all kind of amino acid derivatives? How would you be able to determine that without knowing specifically that it is an amino acid?
Have nitrogen group.
Yeah, very good. The presence of nitrogen. When you start seeing nitrogen, you can think, well, maybe we're a nucleic acid group or we're a amino acid group because nitrogen is present in those. It's not present in inositol, right? We know inositol is a carbohydrate. Okay. No glycerol in that one. All right, so the amphipathic molecules are what make up the cell membrane in usually a double membrane manner. right? They line up tail to tail and then the heads squished together on from side to side. You know, it's just a hydrophobic, hydrophilic interaction. It's not a covalent bond. And that's why membranes are very fluid. The phospholipids can move throughout the membrane, right? It's a very fluid system. All right, lipids also involve glycolipids. So these are sugar and lipid combined, right? These are the molecules that associate with membranes, are also involved in post-translational modification, and they're also amphipathic, right? So they will have differences in polar differences across the molecule. So when I say steroids, what do you think of when you hear steroids? Schwarzenegger, right? Anabolic steroids. Well, there's a reason for that because steroid lipids are a different group of fats that contain these steroid rings in them. And the most commonly understood studied steroids are the masculinizing and feminizing hormones, testosterone and estrogen. So these don't have structural roles in the cell, but they have messaging roles, right? They're important in chemical messaging. You know, the system of generating these steroid lipids, these hormones, and then delivering them to the body to have effects on the cells. We're going to talk a lot about cell signaling towards the end of the course. But all of these steroid hormones come from a basic steroid structure called cholesterol. You got good and bad cholesterol is the good cholesterol we're talking about. We need it to make these steroids lipids. It's a precursor molecule to build these different lipids. All right, this is sphingomyelin. Oh no, this is glycolipid here. Right, so the glycolipids are polar. Important to know that. Got that glucose group up there, which makes that whole presence of that oxygen there makes it polar. All right, that's it for lipids. Moving on, nucleic acids. It's a little easier because there's only two types of nucleic acids, DNA and RNA, but the way that they're built and the way they work is a little more intricate. DNA stands for deoxyribonucleic acid, which means it has a sugar part, which is the deoxyribose. It has a nitrogen-containing basic region that is also attached to that. And then in order to stick nucleic acids together, you have to have a special type of bond called a phosphodiester bond that contains phosphate in it. So the typical nucleotide, right, will contain phosphate, the sugar group, and the nitrogenous base. The other term you'll see often is a nucleoside, which is basically the nucleotide minus the phosphate. So it's just the sugar group and the nitrogenous base. Within DNA and RNA, it's that nitrogen-containing base that varies and makes the different nucleotide, whether it's A, T, G, or C. So we say DNA has two nucleotides, the A's and the G's, which are in a family called purines. And those are the larger nitrogen-containing bases. And another family called pyrimidines, which are the C's and the T's, which are the smaller nitrogen-containing bases. They're all attached to deoxyribose, though, and then ultimately a phosphate as well if we're talking about a nucleotide. All right, DNA and RNA are the same with some subtle differences. So RNA has a different sugar group. It has ribose instead of deoxyribose. And it actually doesn't have any thymine that exists. Instead, it has uracil, which makes it different. Additionally, typically, DNA exists in double two strands, and RNA exists single strand. Right? You should know these families well, purines and pyrimidines. Purines are larger, they have two rings. Pyrimidines are smaller, they have one ring. But if you notice, A's to T's, C's to G's, you always have purines hydrogen bonded to pyrimidines as a result of complementary base pairing. All right, so we can start down here. You can see the nucleotide has the deoxyribose sugar group. Ribose is RNA, sorry. The nucleotide has the ribose sugar group, it has the nitrogen containing base, and it has the phosphate. All right, if you're talking about a nucleoside, it doesn't have that phosphate. It's just the sugar and the nitrogen containing base. If you've got deoxyribose on, you've got a DNA molecule, and ribose goes to RNA molecules. The purines are the larger nitrogen containing regions, having 2 rings each. And the pyrimidines are the smaller regions, having only 1 ring. RNA doesn't have thymine. Instead, it has an alternate nitrogen containing base called uracil, which is also a pyrimidine. Okay? Purines bond to pyrimidines. Purines are the A's and the G's. Come up with some sort of little acronym or something to memorize. All right. If I shared you mine, I would probably get arrested. So keep those to myself. Oops. All right. So as nucleic acids polymerize, right? So you basically have nucleoside, phosphate, nucleoside, phosphate, and so on. What happens is the way the ring is structured on the ribose or the deoxyribose, the phosphate group is attached to one specific carbon, and the hydroxyl group on the other end of the molecule is attached to a different carbon, right? The phosphate is attached to the 5' carbon. I'll show it on the next figure. And the hydroxyl group is 3'. Memorize this, right? Memorize this well. So it gives the molecule polarity. If we have one strand of DNA on one end, you have that phosphate sticking out, so we call this 5'. On this end, the hydroxyl is sticking out, so this is 3'. So it has like a charge difference. And the way that in a double-stranded manner, DNA molecules will line up is this in this complementary fashion, where on one end you have 5' here, that means you have to have 3' here. Okay. It's going to be really important when we talk about transcription and when we talk about DNA replication, how the molecule is situated. Right? How does polymerization occur? Through the production of these phosphodiester bonds. That bond is very energetically unfavorable. So you need to have a way of providing energy to it. And the way that that's done is the availability of these things called DNTPs. Dinucleotide triphosphates, right? So a phosphodiester bond only has 1 phosphate in it. But what it's adding on to the molecule, the nucleoside, comes from a DNTP, which is right here. Having those 3 phosphates attached to it provides enough energy to make this reaction energetically favorable. Okay? Whenever you're doing PCR in a reaction tube, right, what do you have? Remember, PCR is a process of what's basically in vitro DNA replication, right? It can amplify pieces of DNA. And when you do that reaction, you have to have DNA polymerase, the enzyme that builds DNA. You have to have some buffer, and you have to have all 4 DNTPs. Right? And they have to have those 3 phosphates on to provide enough energy to produce the phosphodiester linkage between, in this case, this nucleotide and this nucleoside. Okay. DNTPs. All right, 2 strands are held together by hydrogen bonds. Now, because because you have purines and pyrimidines, right? You have nucleotides that have 2 rings and some that have 1 ring. You can kind of do something with the hydrogen bonding. Right? So if you look here, when you have C's hydrogen bonded to G's, so this is one strand of DNA here. This is the other strand, and they're stuck together through hydrogen bonding. When you have C's and G's, you have 3 bonds present. But T's and A's only have 2 hydrogen bonds. Right. Which one's stronger? CG hydrogen bonding or the AT? Yeah, there's more bonds. If you have a sequence that is CG heavy, right, it takes more energy to unzip that molecule, right? It takes a higher temperature if you're doing it in vitro. And because the molecule has a higher melting or boiling point, is the presence of those triple hydrogen bonds between C's and G's. All right. Remember, like we said before, it's this complementary base pairing that enables processes like transcription and translation, a flow of information, and also enables DNA replication, right? Because we can split the molecule, break the hydrogen bonds, and we have a template on one side. By reading just that side, you know what the other side is because of complementary base pairing. All right. So, this is not the final structure of DNA, but all of the hydrogen bonding and the polarity of these phosphates in solution, it will cause it to wind up and twist up into its final conformation, which is like a spiral staircase, or also called the double helix. The structure of the double helix is something we're going to get into a lot because it has a lot to do with how molecules come and interact with the DNA. Because if you look, it's not a completely symmetrical molecule. There are these large grooves, which are called major grooves, and there's smaller grooves, minor grooves, and how molecules interact with that DNA are dictated a lot by that UV and stuff. Okay. All right. So, like I said before, RNAs probably the first informational molecule in biology because they can be enzymatic. And one reason that they can carry enzymatic activity is their ability to fold up into these weird shaped molecules. And it's, shape determines function. So if it folds into some weird shape, do that. So it can actually self-base pair with itself. Self-base pair with, that doesn't sound too good, but C's to G's and U's to A's, because remember, there's no T, right? Typically RNA is considered a single-stranded molecule, but this is one piece of RNA that is folded up on itself. That's the difference between DNA and RNA. Like DNA typically won't necessarily do that, although it can in some circumstances, depending on the sequence, but RNA can readily fold up on itself and form these different conformations. This is a specific type of RNA called a tRNA or transfer RNA, which, as we'll see later on, is a very important molecule in guiding the production of proteins, right? So it's the tRNA which gets charged with an amino acid and comes into the ribosome to cause proteins to polymerize. All right, so transcription and translation and DNA replication, all capable because of this complementary base pairing. You can see in DNA replication, it comes apart, right? And each strand that's come apart can act as a template on how to build a new strand of DNA. With transcription and translation, the same thing. It comes apart, and now only one side of the molecule needs to be read, and that can be used to generate an RNA, or the RNA, if it's being read, can be used to generate a protein. All right, getting close there to the end. The last group are proteins. All right, so these are the functional molecules of the cell, really. And, some carbohydrates can have some sort of structural functions, and certainly RNA has its ribozyme activity. But when you're looking at the molecules that actually do things in the cell, it's your proteins. Not only do they make up structural components like the cytoskeleton and things like that, proteins can be broken down to process energy and extract energy from, but proteins are the signaling molecules and the enzymes within a cell. They can transmit information between cells. You also have protein hormones, right? So we talked about the steroid lipid-based hormones, but a hormone like insulin, for example, is a protein hormone. Proteins make up antibodies. Antibodies are important in immune defense, and also binding to other molecules. So whereas nucleic acids are only made up of 4 different nucleosides, or 4 different nitrogen bases. Proteins are made up of amino acids, and there's a greater diversity of them. There are 20 different amino acids. They're all grouped according to whether they're nonpolar, polar, basic, or acidic. And it's all what decides the chemical properties of that amino acid is a specific group of the amino acid called the side chain. And you see a figure that explains that. Now, on your exams, most every exam you will have a figure that has all the amino acids on it, okay? Just so you know. So you'll be able to look at the chemical characteristics of the amino acid and kind of make your decisions on exam questions from there. Biochemistry, probably wouldn't have that, memorize the structures, but we're not going to do that in here. All right, so here's a generic figure of a amino acid. Amino acids have a central carbon, a little hydrogen sticking off to make 4 bonds. But it has a COO negative or carboxyl group. It's an example of a functional group on one side. We call this side the C terminus of the amino acid. And it has an amino group, which contains nitrogen on the other side. It's positively charged. And we call that the amino terminus or amino end. All amino acids, all 20 of them have this structure, but where the 20 are different are in that side chain. That's what dictates what amino acid it is, what group it goes into, if it's polar or nonpolar. And yes, that's it. Okay, so there's your 20 amino acids. Most of them are just nonpolar amino acids. All right, now if I were to ask you, if you have a 3D structural protein here, where would you find most of the nonpolar amino acids, like in the center of the protein or in the exterior of the protein? Center, why? Hydrophobic, because they're nonpolar, right? They don't want to interact with water. You're going to have all of these polar amino acids here more towards the exterior. Polar amino acids there towards the exterior. Okay. Again, this figure will be given for you. You need to start to become familiar, though, with all these terms, right? You should know if I'm talking about phenylalanine that we're talking about an amino acid. Get familiar with the 3-letter abbreviations as well, because those are readily used. But again, this figure will be provided for you on the exams if necessary, okay? All right, so just some crucial things, and it looks like some of you guys already have it figured out. So the nonpolar are the hydrophobic amino acids. They'll typically make up the interior of a protein. Specifically, 2 of them, the cysteine and the methionine, contain sulfur groups in them, and they can actually produce what's called a disulfide bond, which can help make a protein into its final conformation, its final shape. The polar are hydrophilic. They're typically found on the exterior of the protein, right? The basic group, these are positively charged and hydrophilic because of their chemical makeup, because of that side chain. They're found on the exterior because they're hydrophilic. They can also interact with other molecules, other amino acids, maybe in the same protein or in other proteins. And then the acidic group, these are negatively charged. And just like the basic group, they can also interact with other molecules as well. They're also hydrophilic. So all of these guys, polar, basic, and acidic, exterior of the cell, nonpolar, which makes up most of them, are found in the interior. And no, remember that there are a couple amino acids that can form disulfide bonds. Okay. Now, how these amino acids grow is a process that occurs during translation of proteins. It occurs on the ribosomes. And the formation of polymerizing a protein is achieved through producing what's called a peptide bond, right? These are 2 amino acids that join together. The carboxy term of one joins to the amino term of the other. Right? That reaction, as you can guess, is energetically unfavorable. So you have to have an intermediate molecule that provides more energy. And the intermediate molecule we've already mentioned is that transfer RNA molecule that actually gets an amino acid tacked onto it. When that's happened, we say that tRNA is charged. And a charged tRNA is ready to come into the ribosome and give its amino acid to produce now the growing protein. All right, so let's look at that a little more detail here. So here we've got the production of a peptide bond, energetically unfavorable. I think this figure maybe gets a little confusing here. So you've got these R1 and R2, and that's indicating like the number of amino acid in the chain, right? So, and look down here to our end product here after we have the amino acid. It's the R1 that's in like the growing chain. Okay, that's what I want you to understand. So like if this is the growing peptide here, and this is what's coming in, this is R1, and this is R2. That's how the thing is being built. All right, so energetically unfavorable. So we're going to have this two-step reaction that kind of makes the whole thing energetically favorable. The first thing is an interaction with ATP. Okay, and you can see what happens here. Not only do you lose a phosphate, right, but adenosine actually gets transferred to that first amino acid, making that conformation energetically favorable. The second thing that happens is a tRNA molecule gets charged, right, and comes in, and now we have a charged tRNA instead of the uncharged one there. Right? And the charged tRNA now can easily give its amino acid to the growing peptide strand. That addition of adenosine, though, is essential in the charging process, right? And that's like step one. And then step two is the release of that adenosine and the replacement with it with an amino acid that's charged. Okay, two-step reaction to drive peptide synthesis. All right, we call this tRNA like an adapter molecule because you've got 20 different amino acids in the cell, so you have 20 different tRNAs in the cell. And the tRNA is nice because a region of it, right down here, you have a region called an anticodon. It can actually bind to a specific sequence on an RNA molecule. So that's how we can transfer information from sequence into determining what amino acid we're going to get. All right, let me just take a look here. It might be a good time to take our last break for the day. Yeah, actually, you know what? Why don't we just keep going and get out of here? All right, let's do it. Oops. All right, so structure determines function. Proteins are complex because they have 4 different levels of structure. Primary, secondary, tertiary, and some proteins are quaternary. A lot of proteins also just kind of end here. They have final tertiary structure, and that's it. The primary is the sequence, right? So it's a sequence of amino acids. Okay, this is thymine, this is isoleucine, this is glutamate, blah, blah, blah. Now, what happens because of hydrogen bonding is some regions of that amino acid chain will fold up independently into one of two specialized structures, which are secondary. We call these beta sheets and alpha helices. Okay, so imagine along that peptide chain, there's a methionine, and now we have a beta sheets. Now we have an alpha helix, and then there's our last amino acid. This is not to scale, obviously there's a lot of amino acids that make up the secondary structures. But you can start to see within the primary structure, now you have the presence of these additional structures, which are beta sheets and alpha helices. And then finally, to make a tertiary structure, this whole thing folds up. Those are beta sheet, those are that, those are that. The worst artist. And see just how this thing is coming together to its final folding, which is going to make its final shape and give it its function. Okay. Oftentimes proteins stop there, but sometimes multiple tertiary structures will come together to form a larger quaternary structure. An example is hemoglobin. Hemoglobin has 4 tertiary structures that are indicated alpha 1 and alpha 2 and beta 1 and beta 2, and they're all joined together by what? Iron. Elemental iron. So quaternary structures are multiple tertiary structures that are stuck together. Okay. The final shape of the protein determines function. So if you mess that shape up, you mess up the function. And that's what protein denaturation is. When you denature a protein, you start to unravel the shape. You know, cooking, heating, providing weird salt conditions or acidic or too alkaline environments will start to affect the shape because you start messing with that hydrogen bonding. And proteins can refold as long as not too much unfolding has occurred, as long as the condition becomes favorable again. But, you know, if you get too far out of that range, you can permanently disravel the protein. Okay. So look at this, for example. This is, I think, insulin, right? It's insulin. So insulin has this really complex process of forming. There's all these post-translational modifications that occur with enzymes that actually take the protein and cut it and then stick it together in these weird orientations. And one of the things that sticks, you can see the 2 different peptides together, is this disulfide bridge, right? And it's the cysteine residues that readily form disulfide bridges to help stick proteins together. Now, like when you're working in a lab, we're going to see that you can heat proteins and unravel them, but the heat is not going to break those disulfide bonds. You have to add an additional reducing agent. So we're going to use chemicals like beta-mercaptoethanol, DTT, things like that to break those disulfide bonds so we can completely break apart proteins. All right. Here's a protein called RNase. It's not RNA, it's a protein. It's called RNase because its job as a protein is to break down or hydrolyze RNA. And you can see it's got disulfide bridges. And you could heat it and treat it with like that reducing agent I talked about, and now you've unraveled it. But if you improve the conditions, right, you can renature the protein. As long as the enzyme is there to produce the disulfide bridge, it will make those disulfide bridges and return the protein back to its native conformation. Oops. All right, so this is a tertiary structure, myoglobin, not the same thing as hemoglobin. Right? It's got a little heme group. This is one tertiary structure. And often proteins are not just a series of amino acids. They will have inorganic molecules associating with them to provide their function. A lot of enzymes have these additional inorganic groups and chemicals like zinc, and zinc's a big one. Heme is a big one, iron, to assist it in its catalytic activity. All right. The secondary structures, the alpha and beta sheet, those are formed ultimately by hydrogen bonds. So you can see all the hydrogen bonds that are sticking this thing together in that alpha helical conformation. And also the beta sheet, also called a pleated sheet, like, you know, the blinds that you see, like the pleats on a pair of pants, sorry. Good example of that sort of shape or conformation. Again, it's hydrogen bonding that's doing these orientations. All right, one important term here, the domain. So tertiary structures, the final folding has domains. And, you know, you can identify the domains in just the sequence of the amino acids. Right, so our protein, right? Maybe this right here is a DNA binding domain, which means it's going to latch onto a piece of DNA. And this right here is the catalytic domain. Maybe it's going to cut a molecule or something like that. Different domains or different regions of the proteins that have different jobs, so to say. And it's really only at this tertiary structure that the domains can function because they have the correct shape. They're typically about, you know, around 100, 200 amino acids long. They are determined by polarity largely, right? So domains that are very hydrophilic are going to be on the exterior of the enzyme of the protein, and domains that are hydrophobic will be on the interior. Right, here's a good example of ribonuclease. You can see ribonuclease has these ribbons that are going through it. Those are indicating the beta sheets and the alpha helices. And then there are just looped regions, which would be like this over here, that are connecting these secondary structures together. Okay, the more complex enzyme over here with more domains. And the quaternary structure, like we talked about, is multiple tertiaries together, usually held together by an inorganic molecule, right? So hemoglobin is a classic example of that. Proteins can also contain intrinsically disordered regions. Okay, this is an important concept to grasp. When you think about like protein biology and protein function, you know, how a protein works is based a lot on its ability to do different things. Let's check something here. So to give it like molecular diversity, obviously we have all those 20 different amino acids. We can build it in different ways and we can do different post-translational modifications. But if we have regions of the proteins that can adopt different various conformations and constantly change, they're intrinsically dynamic. Now we can really start doing some different stuff. There's a really good example of this when we get to the nuclear pore complex, where you can see all these intrinsically disordered regions that go into the middle of the pore, and it can help decide, you know, if molecules can get in and out of the pore. But a lot of this is still, you know, not completely understood how this works, but they've been readily identified, and we know they have important purposes. But you know, how this level of disorder really contributes to biology is, we know, is very significant. And there's some randomness associated with it, but it's probably not all random. And it just remains to be completely understood. But you can see here, you've got ordered regions, right? And then you have regions that can change, that can adopt different conformations. Maybe it gives this protein two different jobs. It can do two different things. When the disordered region is one conformation, it can do one thing. When it's in a different conformation, it can do another thing. So physically disordered proteins make up intrinsically disordered regions in larger proteins that can ultimately just be structured and not have any intrinsic disorder to it. Okay. I know that was a lot for the first day. Again, we only meet once a week, so you know, expect a head full each time we get together. It's a real pleasure to meet all of you. I really look forward to working with you this semester. So I'm real caffeinated next time, and try to study this information a little bit, because I'm going to give you that mock exam, which is just going to take 10 or 15 minutes, you know, just to give you an idea, just for yourself, where you sit and how much work you think you're going to need to put into this course. Okay. You guys have a wonderful week. I'll see you Monday for lab. Just coat all those lab notebook.
Yeah, what about gloves?
You got gloves. You're welcome. Oh, shoot. Didn't take attendance. Whoops.
Everybody was here for the day, you'd be, yeah.
I'll put something on Canvas. You guys will have to just log in. Whoops. Yes, ma'am.